To be able to observe structure and structural changes of RNA in living cells is really what gets me up in the morning. And the methods we are developing nowadays in the lab really are the first step to actually allowing us to do so.
RNA, the overlooked molecule that transports information from the DNA molecules to the proteins, is what Katja Petzold's research group focuses on.
The dogma in biology says you have DNA, where the information is stored. It's a very stable molecule, so you can see DNA in dead bodies for 50,000 years. RNA is very unstable. It's made to just transport the message, the short-term message, to make a protein. But a lot of theories say that the RNA was there first, because RNA is actually very versatile. It can store information. RNA viruses, for example, have this, similar to DNA, but it's less stable. RNA is really a central part, and it's very little understood. Protein has been studied like crazy, because it's the actor, and you want to change something, you change the actor. And the DNA has been studied just to understand, okay, where are coming our inheritance from our traits. So a lot of things have been understood, but because it was just thought, okay, this is a short-lived molecule transporter, it wasn't super important. But now it's shown that, okay, there is much more importance to this molecule, much more regulatory and also activity present in this molecule. We actually need to study and understand it.
Since a lot of RNAs are drug targets and RNAs are also used as drugs, understanding their structure and movements in the cell will increase drug efficiency. The reaction of structured RNA to the environment isn't yet understood, and by observing it up close, answers to why the drugs sometimes don't work can finally be given.
We study RNA structures and dynamics in living cells in order to understand how the environment of a cell does change the RNA structure and dynamics, and be able to predict and manipulate those with drugs or molecules or just mutating everything.
The team prepares RNA molecules in their wet lab. Here they're studying RNA molecules in water and have recently started to observe the structures of RNA in living cells.
Here we are in our RNA wet lab. This is where we produce RNA samples. We produce them biochemically in a reaction overnight. We put the building blocks in a tube and make them. This one is a sample I just prepared for the gel for loading it later, so we actually make it colorful to be observable.
The samples are then brought to the NMR room where the team can observe the molecules and their reactions to different scenarios.
We are now in the NMR room, where our actually big magnet is sitting here. It's an ultra-cooled magnet, so inside there's like 4 Kelvin temperature, like really, really cold. And we're gonna put the sample we quality controlled this morning now in a magnet.
NMR stands for Nuclear Magnetic Resonance Spectroscopy, and the NMR machine can see tiny structures in the cell. The team later examines the results to study the molecules up close. This will provide much-needed answers to how medicines could better target the molecules and other questions the scientific community is looking to solve.
There's very little research done. So there's not so much to compare with and not so much to already read up on. So we still have so many open questions. Sometimes we don't know where to start first. Better research in this area can lead to a lot of things. Our prime target is to understand regulation better and find ways to stop the system what it's doing, or using it as a drug. It means enhancing what it is doing. Really, you can do both. The dream goal of our research, I want to be able to say we can measure this drug in a cell from a patient and tell the patient this is why it doesn't work, that's how we can change it. We often don't know why drugs in cancer therapy stop working. We don't often know why antibiotics don't work. But with the method and the work we do here, we hope in the future we can answer this question.